Good morning, everyone. Today we will start the part number 2 of chapter number 5. Today's topic will be procedure of hybridization. Whenever we are going to use the hybridization technique, then different procedures are followed. Out of that, the first one is selection of parents. We will select the parent according to our objective and as per demand. After that, we will follow the hybridization techniques. There are different hybridization techniques in which emasculation, the second one is bagging, the third is anthesis, the fourth is tagging. In general, Gregor John Mendel used the garden pea plant for his hybridization experiment, and he performed the experiment up to F3 generation. After following all these techniques, crossing is done, and finally we will harvest and process the product. So let's see the procedure of hybridization. So in the procedure of hybridization, you can see the figure. There are 7 steps are involved in hybridization. The first is choice of parents or selection of parents. Second, evaluation of the parents. Third is emasculation, bagging, tagging. After that, pollination, harvesting, thrashing, drying and storage of seeds. So this figure is taken from your book, and some steps are added here. In the first one, pollen, male sex organ is cut off or removed. This is known as emasculation. Emasculation means removal of anthers. In the second step, pollen of white flower is taken and transferred to the pistil of the purple flower. This is known as anthesis. And in the third step, the fertilized pistil gets mature and becomes pods. In the fourth one, seeds in each pod are ready to be planted, and this is the new generation which is grown in the fields. Process of hybridization, the first step is selection of parents. Male and female parents are selected on the basis of characters. So we will choose the character, what type of parents we want to use or we want to use in the experiments. So this will be the selection of the parents. In the second step, emasculation. This is important and it is also the part of practical examination. What is emasculation? Removal of stamens or anthers or killing the pollen of a flower without the female reproductive organ is known as emasculation. In bisexual flowers, emasculation is essential to prevent of self-pollination. So it will protect the flower from self-pollination, and in monoecious plants, male flowers are removed. So you can see this is the anther which is cut with the help of scissor or removed with the help of forceps. This process is known as emasculation. In the bagging is a process used in plant breeding to prevent self-pollination in bisexual flowers. So this is the cover. You can see these are the flowers, and these flowers are covered by polythene or any cloth or paper. So this is the bagging process. Flower is covered by a cloth or polythene. Flower is covered with a paper bag to prevent contamination from unwanted pollens. So it will prevent the flower from unwanted pollen. The next is anthesis. So anthesis is the period during which flower is fully open and functional. It may also refer to onset of that period. So the transfer of desired pollen on the stigma of the flower is called anthesis, and brush is used for this purpose. So you can see a close picture of wheat inflorescence, wheat head flowering anthesis. Notice the anthers sticking out. So you can see these are the anthers coming out from the flower. Next step is tagging. The attachment of tag to identify the plant is called tagging. So you can see tagging is the process in which we will use a tag to identify the plant here. Now next is types of hybridization. As far as types of hybridization is concerned, this is of different types. First is intervarietal hybridization. The parents involved in hybridization belong to the same species. They may be of two strains, varieties or races of the same plants. So species is same but variety is different. The next is interspecific hybridization. Cross between different species. We will use the different species for crossing. Intergeneric hybridization between different genera. Here genera will be the different, and the next and last is distant hybridization. This includes crosses between different species of the same genus or of the different genera. Here it may be the different species or it may be the different genera. This is distant hybridization. The next, there is a question, what do you mean by the pure line? What is pure line? So a pure line is descent theoretically reliable from the inbreeding of completely homozygous and comparable parents. Basically, when you want a pure line, then we will follow the self-pollination. So pure line is a self-pollinated descent of a self-pollinated plant. Pure line varieties are homozygous individuals having two of the same allele, whether dominant or recessive, and the homogeneous, same or similar kind of nature. So you need to know the only pure line. What is pure line? It is obtained from self-pollination. Alphabets used here, you can see capital letters used for the dominant trait expressed at the F1 stage. So we will use the alphabetical representation of the plant, in which suppose there is a tall plant which is dominant character, it is shown by capital T, and if the plant is dwarf or you can say recessive character, it is shown by small t. Capital T and T are the alleles of each other. Hence in plants the pair of alleles for height would be capital T or capital T or small t. You can see there are the three possible genotypes. Capital T it will be a tall plant, capital T it will be a impure tall plant, and this is the dwarf plant, and recessive character is always pure. So you need not to write the pure word here. In a true breeding, tall or dwarf P varieties, the allelic pair of genes for height are identical or homogeneous. So in the true breeding, the product will be homozygous, and this is capital T, small t respectively. So capital T and small t are called genotypes. So these are the genotypes and both are homozygous. Capital T small t plant is heterozygous for genes controlling one character, and here that character is the tall or height of the plant. Descriptive terms tall and dwarf are the phenotypes. I have already explained what is the phenotype, so I need not to explain again here. It can be observed outside. Now come to the point. Next, inheritance of one gene. So the study and analysis of inheritance as a result of monohybrid cross is called one gene inheritance. Monohybrid cross means when you have selected a single trait or character for the hybridization, then it will show the inheritance of single gene or one gene. Basically it is based on monohybrid cross. So again you can see the figure here. Monohybrid cross between tall and dwarf varieties of pea plants is as follows. So you can see these are the parental generation. You can see this is the tall plant, capital T. This is short plant or dwarf plant, small t. In the gametes, these alleles are separated, capital T and small t, and after the crossing, or you can say after fertilization, capital T small t is formed. This is F1 generation, and F1 generation, you can see here genotype all are capital T small t and phenotype all are tall plants. So all the plants in F1 generation will be tall. When these plants are undergone self-pollination or selfing, then you can see we will obtain the F2 generation here. These are the gametes capital T small t of the tall F1 plant and capital T small t of tall F1 plant. When these are crossed here, capital T, this is the tall, capital T small t, this is tall, capital T small t, tall, and these upper three plants are tall in nature, while as there is a single plant which is short. You can see here these three plants are tall while this is the short plant. So phenotype is tall is to dwarf is 3 is to 1 and the genotype is 1 is to 2 is to 1. Again, we will discuss it in detail here. Monohybrid cross. What is monohybrid cross? A monohybrid cross is a cross between two organisms with different variations at one genetic chromosome of interest. So we will select a single character here and its phenotypic ratio will be 3 is to 1 and genotypic ratio will be 1 is to 2 is to 1. Again, you can see the monohybrid cross by having a different character here, a cross between a pea plant that is homozygous for yellow seeds. You can see this is yellow seed dominant character, while this is the green seed which is recessive character. And you can see the F2 generation here, capital Y small y, sorry, this is F1 generation here, and in the F1 generation all the seeds are yellow in nature. Similarly, again one figure is given here. This is the tall plant, short plant. I have already explained, nothing to say different from above. You can see tall, capital T small, capital T, capital T small t, capital T small t, small t, small t. These three are tall and this one is short. And again the phenotypic ratio is 3 is to 1. There is a question for you. A pure round seeded pea plant is crossed with wrinkled seeded pea plant. Find out the genotypic and phenotypic ratio in F2 generation. Then phenotypic ratio will be 3 is to 1 again and genotypic ratio will be 1 is to 2 is to 1. Here round is shown by capital R and small r. Capital R is the symbol of round and small r is the symbol of wrinkled allele. So when you will undergo the cross, this is the F1 generation where all seeds of the plant will be round, and in F2 this will be the result. Again, a pure round seeded pea plant is crossed with wrinkled seeded pea plant. Find out the genotypic and phenotypic ratio in F2 generation. So then you can see the above figure there. Now mathematical analysis. This is very important, that the mathematical analysis of the monohybrid cross, the genotype of monohybrid cross is shown by binomial expression as shown below. Genotype is capital T, capital T. There is a single plant in the Punnett square. Two plants are capital T small t, one plant is small t small t. So you can see 1/4 tt, 1/2 capital T small t, 1/4 capital small t. By applying binomial expression, when you will apply the binomial expression a plus b to the power 2, a square plus 2ab plus b square. So here we are going to express capital A is ax and b is by. Then ax plus by to the power 2 equals to ax to the power 2, 2ax by plus by to the power 2. Here small a is half, small b is half, x equals to capital T, y equals to capital T. Then again we will receive the above equation. 1/4 capital T, 1/2 capital T small t, 1/4 small t. And the final ratio, genotypic ratio will be 1 is to 2 is to 1. Thus it is explained by mathematical binomial expression. Now we will discuss about the a new cross, that is test cross. What is test cross? It is introduced by Gregor Mendel. So you will see it involves the breeding of an individual with a phenotypically recessive individual. So we will cross the specified individual with a phenotypically recessive individual in order to determine the zygosity of the former by analyzing proportions of offspring phenotypes. We will find out the zygosity here when F1 hybrid is crossed back with the recessive parent is known as test cross. It is used to know the genotype of the given plant or animal. So you need to memorize these two. Monohybrid test cross is 1:1 and dihybrid test cross is 1:1:1:1:1. You can see, suppose these are the two sheep given here, unknown dominant phenotype. We don't know what is the phenotype here. Either it is pure black or impure black here. Pure black stands for capital B, and impure stands for capital B small b. This is the white animal. You can see non-homozygous recessive small b. This is confirmed, this is recessive. So what will you see? If unknown is homozygous, suppose this animal is capital B, and when you will cross with this animal, then you can see all are black. So phenotypic ratio is 100% black. It shows the pure form of the phenotype. On the other hand, suppose this is impure capital B small b and it is crossed with this animal, then you can see 50% black and 50% white. It is the symbol of impurity of the blackness. Again, a question is given here. A pure purple flowered plant is crossed with white flowered plant. We are going to cross the purple flowered plant with white flowered plant. Its F1 generation is crossed with its recessive parent. Its F1 generation will be capital W small w, and it is crossed with white flower plant. Then find out the test cross ratio and its zygosity. So this is the diagrammatic presentation of test cross. This is homozygous recessive plant, WW, this is WW, and this is the homozygous recessive. So we will start from here. You can see there are two chances of purpleness. This purple is pure purple and this purple is impure purple. Capital W, capital W, it is crossed with small w. Then what will you see? In the F1 generation, all flowers of the plants are purple in color. So result, all flowers are violet or purple. Interpretation, unknown flower is homozygous dominant. If dominant character is arised, then it will be the symbol of purity. On the other hand, here half of the flowers are violet and half of the flowers are white. So unknown flower is heterozygous here, capital W, small w. It is very important test cross based on monohybrid cross. Now there is a Punnett square. Basically, when you are going to draw a tabled format for the genotypic and phenotypic expression, then it is called Punnett square. It is devised by Reginald C. Punnett. The Punnett square is a square diagram that is used to predict the genotypes of a particular cross or breeding experiment. The diagram is used by biologists to determine the probability of an offspring having a particular genotype. When you are going to show these gametes and these are the offsprings in a tabled format or in the square format, then this is known as Punnett square. Now the next is principles or laws of inheritance. Originally proposed by Gregor Mendel in 1865 and 1866, and it was derived in the form of laws by Karl Lorenz. So basically it was changed in the form of laws by Karl Lorenz. So this is to remember. Mendel proposed two general rules to consolidate his understanding of inheritance in monohybrid cross. These rules are known as principles or laws of inheritance. There are two important laws based on monohybrid cross: law of dominance and law of segregation. This is also known as first law of inheritance and second law of inheritance. So we will start the point from law of dominance. When two alleles of an inherited pair is heterozygous, then the alleles that is expressed is dominant, while which is not expressed is called recessive. Suppose I have capital T and small t. So in the capital T and small t, the plant will be tall. So dominant character is expressed, while small t is not expressed here, that is recessive. Characteristics are controlled by discrete units called factors. The units which controls the traits or characteristics are known as factors. Factors occurred in the pair. In a dissimilar pair of factors, one member of the pair dominates, that is called dominant, and the other is recessive. Expression of dominant allele in heterozygous condition is called law of dominance. It means in the heterozygous condition, the character which is expressed is the dominant character and it will show the law of dominance. Example, heterozygous tall pea plant, i.e. capital T small t, where T is expressed means dominant while small t is not expressed means recessive. So you can see the law of dominance is used to explain the expression of only one of the parental characters in a monohybrid cross in the F1 generation and the expression of in the F2 and explains the proportions of 3:1. So there are basically three types of dominance, that is complete dominance. This is also known as Mendelian dominance. Mendel's experiment was based on complete dominance. Incomplete dominance, this is the exception of complete dominance, and codominance is also the exception of complete dominance. So we will start with the complete dominance. It is already explained. Have a look again here. Complete dominance is a form of dominance in heterozygous condition wherein the allele that is regarded as dominant completely masks the effect of allele that is recessive. So what will you see when an allele is masking the effect of another allele, that is the dominance? So the Mendel's experiment on pea plants were based on complete dominance. Complete dominance, you can see here, this is the red color flower, this is white color flower. As you will cross these two flowers, if the color of the flower is still red, it means the red color is dominant here and it is showing the complete dominance. Incomplete dominance or partial dominance, this is important. It was proposed by Carl Correns in Mirabilis jalapa. The another name of this plant is Gul-e-Bas. And the another plant which is used here, that is Lathyrus odoratus. One more is Antirrhinum. It is also known as snapdragon or dog flower plant. A heterozygous condition in which both the alleles at a gene loci are partially expressed. Here genes will be expressed partially and which often produce an intermediate phenotype is called incomplete dominance. Here the genotype will be of intermediate pattern. It shows the similar phenotype and genotypic ratio 1:2:1. Have a look here. Example, when a red flowered capital R Mirabilis jalapa plant is crossed with white flowered small r plant, find out the phenotype and genotype in F2 generation. Again, some facts are given here. Incomplete dominance. Incomplete dominance, also called partial dominance, occurs when the phenotype of the heterozygous genotype is distinct from and often intermediate to the phenotype of the homozygous genotypes. Example, snapdragon or 4 o'clock plant or morning glory. This is also known as primrose. A child with wavy hair as a result of one parent's curly hair and other straight hair. And another example is Tay-Sachs disease. Have a look here. This is Mirabilis jalapa. You have taken the capital R, capital R. This is the red flower. This is white flower. When you are going to cross it, so in the F1 generation, you can see all flowers are pink in color, and the pink color is intermediate color of the red and white. Similarly, if we use this in the Punnett square or checkerboard, then this is also capital R, W, W. We can write the W also in place of small r. The colors are pink for all the plants. Incomplete dominance, inheritance in which an active allele does not entirely compensate for an inactive allele. The result is an offspring that is an intermediate between two phenotypes. Already I have explained, and this is the F2 generation where one flower will be red, another two flower will be pink, and one flower will be white. So genotypic ratio is 1:2:1. Explanation of the concept of incomplete dominance. How will you explain this concept? Every gene has a particular information to express the trait in a diploid organism. There are two copies of genes are formed as a pair of alleles. The alleles may be identical or non-identical. That can show the characteristics. The gene can produce two allelic forms. If normal alleles are present, then it will produce normal enzyme which are responsible for the normal phenotype, and it causes dominance. So suppose there is the capital T, both are normal alleles, it will produce normal enzymes, and after the result and after the activity of the normal enzymes, there will be the expression of normal phenotypes. On the other hand, if heterozygous condition is present, then there will be the following chances. In the heterozygous condition, effect of, this is the heterozygous condition, capital T small t, you can see the effect of T allele and effect of small t allele. Capital T allele, this will form the active enzyme. It will be responsible for expression of normal phenotypes, and it will cause the dominance. On the other hand, you can see small t allele, this is inactive or absence of enzyme, and it will disturb the dominance. So you can see again here there is the red color flower and white color flower, and when it is crossed in the F1 generation, you can see capital R small r, capital R small r, capital R small r, and this is the F1 generation where all the offsprings are pink flowered. So you can see the expression in heterozygous condition. So this is all about the incomplete dominance. Now we will start the codominance. Codominance means co means together or coexist. Codominance is the type of dominance where the offspring shows similarity to both the parents, and it is due to the blending of alleles. You can see in the codominance there will be the blending of alleles. Both the alleles will express themselves. Means in this dominance, alleles of a gene pair in heterozygote are fully expressed, thereby resulting offspring with a phenotype that is neither dominant nor recessive, because recessive character is also expressed and dominant character is also expressed. Some example is given here: skin color or coat color of the cattle. So we will see in the cows here, and human blood group ABO system. So have a look. This is the cartoon explaining. You can see this is the blue colored bird CBB here, and this is the another bird that is the white. And you can see some part of this cartoon is still blue in color. It means blue color is expressed, and this part is white in color, so you can see white color is also expressed. When both the alleles are blended, then this is the codominance. One important example given here: when a white cow is crossed with red ox, find out the phenotype and genotype ratio in F2 generation. So what will you see? This is the white cow, RW capital, and this is the red cow, RR, and when it is crossed, this is the roan cow. This is the roan child. Roan means it has both the colors, white colors, red color, white color, red color, white color, red color, white color, red color. It means you can see in our language this is chitkabri. Get it? In human would be the ABO blood group where alleles A and allele B are both expressed. So if an individual inherited allele A from their mother and allele B from their father, they have blood type AB blood group, and the AB blood group is an example of codominance. A figure is given here. You can see there are 4 type of blood groups in the ABO system: blood group A, blood group B, blood group AB, and O. So red cell type will be just like this: antibodies in the plasma. For the blood group A, there will be the antibody anti-B. For blood group B, anti-A. And for the blood group AB, there is the absence of antibodies. And for blood group O, both type of antibodies are found here. And as far as antigens in RBC is concerned, it is just opposite to the first one. And A antigen is present, then group A, B antigen is present, then group B, and A and B both the antigens are present, then AB blood group. And if there is the absence of antigens, then there will be the blood group O. Again, you can see there are 6 possible genotypes are found in human of ABO system. Blood group O is universal donor while the AB is the universal recipient. You can see blood group A, or you can say blood type A, it contains 2 possible genotypes: IA, this is the homozygous, and IA small I, this is the heterozygous. In this condition, AO, and it will be responsible for the blood group A. It can receive the blood from blood group A and blood group O. For the B, again there are 2 genotypes. For the A, B, there is the single genotype, and for A, there is a single genotype here. So, O blood group can receive blood only from O, while AB is the universal recipient. It can receive the blood from A, B, AB, O, all. As far as inheritance of human blood group or blood types is concerned, our blood type is inherited from our parents. Each biological parent donates one of 2 ABO genes to their child. The A and B genes are dominant and the O gene is recessive here. Suppose a possible genetic diagram for inheritance of blood group A. Suppose this is the blood group A, that is heterozygous AO, and this is heterozygous BO. These are the gametes AO, BO, and after the crossing you can see AB blood group type will be AB, BO, B blood group, AO, A blood group, and this is the O blood group. So dear students, we have finished the first law of dominance. Here, tomorrow we will start the second law based on monohybrid cross, that is law of segregation, which is also known as law of purity of gametes. So try to memorize this, try to understand all the facts that I have taught you today, and if you have any problem you can ask to me. Thank you very much. Have a nice day.